A very good evening, class. Warm welcome to everyone for today's second day of a session in the series of Principle of Genetics. Um, I hope you're all doing fine, staying safe, and having a good time back at home. And yes, keep smiling. That should be our motto for life. That's keep spreading smile to others also. When we're done with the lectures today, give it to your parents, give it to your friends, give it to all your friends. Okay, good questions. Uh, so please recommend us some free journals. So, बच्चे free journals are not so good actually, but some of the students have recently published, uh, like in the ICJIRT, something with the ICJE, ICJIRT, IGRT maybe. Like this one, International Journal of Engineering and Research and Technology. So some people have published in them already, and also many others. But their impact fact paper, their recommendation is not so high. But you have to look for in which you can publish. Because some journals will take your reviews within 24 hours and they will publish. But their preference will be not so high. Their impact might be not so high. So so don't use any references from Wikipedia. They are not good one. Within Wikipedia, look where you can find publications like this. So these are not accepted one in the references. So they use lot of Wikipedia stuff there, and keep it same. Like it should not have underline in one case, in others it's not. So your presentation for references should be equal, and you can check it at the end note when you're using the software. Your all things should be the same. So these were some of the mistakes that were happening in it. Uh, there are so many messages that I cannot reply sometimes, and I forgot like who was asking what. So there was one person who just sent me, I think this one. So they publish in the IRJTC. So it was accepted, International Research Journal of Modernization in Engineering, Technology and Science, IRJ So it is published. So they were all bachelor students or master students, and with the help of they put my name there somewhere, and IIT Kharagpur also, Edufabrica, all they were acknowledged, and then everything is done. It's good to go. So maybe I can share with you this one. So VGF, some of the journals from my MBBT students who have published successfully. So I will share in the group. And what else? Um, so I shared one that RG. Yes. That was RGNTS. It's not bad. Something you will get it. Mhm. Any other questions, students? And please like, subscribe, and share this YouTube channel. It will be really great for me. This journey has been started with my YouTube. It has been great. Also, my journey with EduFabrica is both has been really exciting and fantastic during this COVID time. So I really want to thank EduFabrica for providing me this opportunity. Uh, so and on top, if you all students also hit like buttons and subscribe, mainly if you subscribe, it will be really helpful for me to run my YouTube channel. Good evening, students. Yeah, assignments has to done alone. So there were some bache were asking. Somebody has opted only for the project, so they don't have to do the review. If they are thinking of doing a review, they can also join some group and can do it. It's not that you are not allowed to do it. You are most welcome to do so, and so you can just go for it if you want to do it. If and assignments are not compulsory, so you can do it on your own. You have to do it on your own, actually, not in groups. So yeah, the review part has to go in the groups actually. Yes, dear. So shall we start or we wait for a few minutes more? So let Yes, you can do internship also on your own, but I will prefer to do it in the groups. You will learn how to manage groups actually. It's always good to learn team working. You will learn a lot of things in that. So I prefer to go for team learning and doing thing in the group. So where you can see that whether you are a leader or you are working under the leader, you will understand your position while working there. So okay, let's start. I think. Not much more students joining us. So they were somewhere on my LinkedIn also. Somebody sent me, maybe I can share with you also that one. Uh, Somebody shared their paper have published. It was on LinkedIn or on the Telegram, I'm not sure. It was a private message. Let me just share, then we start the lecture. This one. No, okay. Anyhow, I couldn't find it. So I will share it later with you all students whenever we have time. Okay, let's start with the lecture then. With the awesome lecture of today. Principle of genetics. So you all had topics for internship, I guess now. I shared it in the evening. So I found these topics. So just I checked some genetics because these are all genetic diseases, all schizophrenia, autism, obesity. They're all related to the our genes part. And also I choose some other topics with the human genetics, gene mutations, on human diseases, immunization. Some general topics were added also, transgenic organisms. So these are just general topics. You can choose also other topics if you like. So this is just to give you a hint that where you should go in the case of choosing your review topic. It will be easy then. So before I start, let me just check. So our group has selected one topic for the paper, how to begin, like what should be the first step. So, bache, you have to read lot of reviews. First, I would recommend if you have chosen topic, let's say cancer and genetics. So you go to PubMed. This is where you can read a lot of research papers, and click on genetics, cancer review. So you had 1040 561 results. So these are the recent publications in 2014. Gene therapy, progress and production. So you read at least first 10 papers. You at least read these 10 papers fully, and from them you start to think then. But you cannot download them, or I think you can. You click on this link then, PMC full test text, then download the PDF version. So here you have your uh, Royal Society Publishing.org, your first paper with the general therapy, progress and prediction. Uh, so in this case, this figure is there, and the subtopics are there. First is the topic, authors, the place, an abstract, then the introduction, then the figures, then subtopics of your reviews, with each like like a story is going on. So each topic has should have a story. Um, so each topic are covered under subheadings, uh, and then talking about at the end about the future perspective. And you can also write little about the conclusions, a summary, what you have written, all in all. So like this, then at the end is your references. So this I have this format I have shared with you. Uh, review. So you should have your title page, title, including your name, date, abstract. It's written there, just go through it. What things should include your introduction, your body, your conclusion, your literature cited. So all these things has to be there in it. So that's how it is. I hope I have answered your question. So like this, you have to go through it step by step. Minimum page 7 for the review or more. No, I mean you can have 8 or 9 also. So it's up to you. It's up to you how much you're willing to do it. The more you do it, the more it's better, but it should be precise. Review thing is to it has to be precise and very well written, so that if somebody doesn't know about that field after reading your review, that person understand, okay, that's other recent updates, that other recent things has happened in this world of thing. So you can do it by going through it. So I think I have answered your questions regarding reviews. Now let's start the topic for today. So that's your next topic now, principle of genetics. So we did yesterday the Mendel's law of inheritance. Now today we will be dealing with the types of gene actions, qualitative and quantitative traits. So let's start. So there could be different types of gene action according to their characteristics. So there could be one which are interaction by correct single characters such as dominant, incomplete dominance, and codominance, and these are called as intra-allele interactions. But if there's interaction between different pairs of alleles influencing the character of an individual, but if they're influencing the other character, that will be known as intra-allelic interaction, or we can say epistatic. So what is epistatic is the gene which are masking the effect of an epistatic gene, and they are called as epistatic gene, and the gene whose effect is masked. So it's like a gene that masking, there is a gene and they're masking its effect, and that gene will be epistatic gene, and gene whose effect is masked, they are called hypostatic gene. So this will be your hypostatic gene, and this will be your epistatic gene, which is covering the allelic effect of that gene. So it's lead to the modifications of normal dihybrid and trihybrid segregations ratio in the F2 generations. The term epistasis was coined by Betson in 1909. Various types of epistatic gene interactions are. So we will discuss some of these epistasis gene interactions, such as recessive epistasis, dominant and recessive epistasis, duplicate recessive epistasis, and duplicate dominant epistasis. So all we will discuss in different formations. So let's start with the duplicate recessive epistasis. In this, what you can see is there is a complementary gene actions are happening. So name itself says duplicate recessive epistasis. So when recessive alleles at either loci, so here the recessive genes which are present, they are masking the expression of the dominant gene. So this is the dominant gene and this is your recessive gene. So they are dominating they are masking the effect of the dominant genes, and this is called as duplicate recessive epistasis, and this is called as duplicate recessive epistasis. This is also known as complementary epistasis. So this is called as complementary epistasis. The best example of duplicate recessive epistasis is found for the flower color in sweet pea. So the example is in the flower color in the sweet pea. So what is happening is here, purple color of sweet pea is governed by two dominated genes A and B. So purple color is given by the A, capital A and capital B, where the recessive gene is small a and small b, which produces white flower. So a cross between purple flower and white flower strains produces purple flower. So uh, giving a cross between give you first purple flower, because here still your purple flower are dominating there. But what happened in the F2 generation, F1 plants are produced, uh, F1 intermitting of F1 plants produced, giving you ratio of 9 is to 7 ratio. F2 is 9 is to 7 ratio instead of giving you 9 is to 3 is to 3 is to 1. So in F2 generation, that generation you were getting 9 is to 7 ratio. Why is it happening? What is the reason behind it? The reason behind it is uh, that the recessive allele A is epistatic to capital B small b. A is epistatic to capital B and small b, and is masking the expression of these genes. And another recessive allele is small b is epistatic to capital and small a also, and they're masking the expressions. So here you can see that after having these genes, we put the law of segregation. We make different combinations A B, small a is capital B, and then small a and small b. Okay, so this is how it is. Then you make a cross between male and female. So as I told you that this small b and small a, if they are both present, so they are presenting the epistatic effect on the dominant gene. So if small b is present, then it is white. If it is small is present, then if it is white. If the small is present, then again it is white. So that's how they are dominating the effect, and you have a different ratio. You have a different phenotypic ratio, that is 9 is to 7. So that's how this complementary gene action gives you 9 is to 7 ratio in this case. Then comes the duplicate gene action, which is when dominant allele at either of the two loci can mask the expression of recessive alleles at the two loci. It is known as duplicate dominant epistasis. So here in previous case, we have a recessive gene who was like taking the case of recessive gene. In this case, dominant gene is masking the expression of the recessive gene. So it's other way around in this case. So in this case, we have a ratio of 15 is to 1. So in the case, for example, on character, like how much is your length is. So it depends upon the gene called capital A and capital B. Presence of these two genes can produce an on character, but if it is they are not present, then you have a homozygous recessive stable state where you have onless condition with a small a and small b. So this is your parent aunt, capital and capital B, and there's a onless, small a and small b. You made a cross, FA1 is a on rise, but when you cross them again with the various gametes, so you have AB, AB, A, B, B, small a, small b. So like this, you also have on the female side, you made a cross, but in this case, what is happening is capital A is epistatic to small a and small b, and capital A develops a own character. So capital A always developing a own character, and another dominant B is epistatic to allele small and small b. So B is also epistatic to small and small b. So these individuals will appear with the own character. So in that's why if the capital and small b is present anytime, you will have an on character, and in the last case you have small a and small b, that will be an online. So that's why you have a ratio of 15 is to 1. So every time your Mendel's law that we were working. So in the type of gene actions, they are working accordingly. So these are the various exceptions to that also. Then there is an inhibitory gene action which is giving ratio of 13 is to 3. In this case of epistasis, dominant allele at one locus can mask expression of the both dominant and uh, second locus inhibitory action. So an example of this type of gene interaction is, example of this is found for the anthocyanin pigmentation, that is in the rice. So what is happening in this color in this case is the green color of the plant is governed by the gene I. So capital I is governed by the green color, which is dominant over the purple color, and the purple color is controlled by the gene dominant gene P. So when we made a cross between I, capital I, small p and small I in capital P colors plant, F1 was green and F1 plant intimating of F1 plants, and we cross the F1 plant again between each other like this. So we have a green plant in 13 ratio and purple plant in 3 ratio. Why so? Why we have 3 purple plants? Because of the presence of the uh, just because they were present in the dominant case. So capital P, and in other case it was green color because the dominant allele was masking it, the both dominant and recessive character. Even though there is dominant present, if there is capital I is present once, so it was masking the effect of dominant and recessive genes both cases. So that's why you have green is to purple, 13 is to 3. Then there is a supplementary gene action. In this, recessive epistasis is happening, giving you a ratio of 9 is to 3 is to 4. Here, one dominant gene has own phenotypic effect, and other dominant gene has no effect. So one dominant gene will be there that will have effects, but other will not. But its own presence with the first gene modify the phenotypic expression. But if it is present with the first gene, it will modify its effect. So thus in supplementary gene, dominant allele of one gene is necessary for the development of the concerned phenotype. So dominant gene, one of the is very necessary so that a phenotype could be present in that case. And while the other gene modify the expression of the first gene, another gene who is dominating is expressing the part of the other gene. So in this case, so capital R, capital P is purple color, and small r and small p is red color. So mostly what we see is here that when we made a cross between them, you have a first purple color with a capital R, a small p, and you made a different phenotypic various gametes ratios like RP, R, capital R, small p, capital R, capital P, and then capital R, small p. And sorry, this should be small r, small p, and this should be also small r. So there are some errors are there, but at the end your ratio is 9 is to 3 is to 4 coming out to be. And then there is an additive factor which is called as polymeric gene action. So in this case, in these two genes, controlling a character produces identical phenotype. So when they are alone, homozygous recessive condition of the other gene. So the both together they are putting the effect it. So if the both dominative genes are present together, so the phenotype effect is enhanced. So A is present, B is present, so A plus B give you enhanced effect of it into your genotypic of effect. So in this, it should be noted that in this case, both genes shows complete dominance. So both genes are showing a complete dominance. So in barley, two completely dominant genes are present. That is gene A and gene B, and they affect the length of the lawn of the awns. Sorry. So the needle-like section of lemma genes A and B. If both are present, then it will be a long awned. If a single is present, then it will be middle awned, and if both are not present, then it will be awnless. So that's how these are affecting. So when both genes A and B are present together, they produce long awn, indicating the effect of A and B on the length added together. So when the homozygous recessive present, it will give you an awnless thing. So here you can see capital A, capital A, capital B, small a, small b. So you have a long awn at the first, because of the additive effect of A and capital P, and then you made different sub gametes, and then what you see is you have 9 long on, 6 on, and 1 onless. So you have a different ratio in this case. Then comes the last and not the least, that is dominant epistasis. So this is an example where we take example of fruit color like white, yellow, and green. So white color is dominated by gene W, and whereas yellow color is dominated by capital G. So white is codominant over yellow and green. So green fruits. So white color is codominant over both yellow and green. So green fruits are produced in recessive conditions, that is WWGG. So cross between plants having white and yellow fruits produce F1 with the white fruits. So here you can see you have a white fruit, capital W, small g, and yellow fruit as uh, small w, capital G. So by crossing them you have a white fruit, and then again you made a cross and you have like this, and then F1 hybrids we have seen that it could be produced in the if we intermate the F ratio, so we see white, yellow, and green in the ratio of 12 is to 3 is to 1. So we saw 12 is to 3 is to 1 ratio, where W is dominant in this case is to small w, and epistatic to capital G and small g. So in this case, so many it's dominating many genes, and that's why we are seeing these kind of effects. So it will mask the expression of capital G and small g. So dominant being dominant, we see 12 ratio in that case. So in F2 plants, we have this WG is 9 is to 16, and W small g is 3 is to 16 types, which will produce white fruit plants. And plant with the WG capital G will produce yellow fruits, and those with the WG and small g produce 1 is to 16 ratio of genotype green fruit plants. This could be also seen in the maize seed coat color and barley also. So that's how it is. So we have discussed many interactions, gene interactions, genetic types of recessive epistasis, dominant epistasis, polygenic epistasis. So now comes a little bit about modifying genes. So here we are discussing about major and minor genes actually. So they are the group of genes which enhances or reduces the phenotypic effect of major genes. Such genes have small and cumulative effect on the expression of major genes, and as a result continuous variation is found in these cases, and which converts your qualitative character into quantitative character. For example, guinea pigs or rabbits, they have piebald spotting. So here you can see in the guinea pigs there is a spotting is happening. This is called piebald spotting, which was produced by the recessive genes when present in the homozygous stage, that is SS. Whenever they are in the homozygous state with recessive genes, we see these kind of piebald spots on the mouse. And degree of spotting depends upon the modifying factors designed as S1, S2, S3. So how much they are present. So enhances and reduces expression of these spotting on the mice. So in the piebald spotting, the modifying factors which produce spotting even in the absence of spotting genes, but their much effect is pronounced in the presence of S. So even though other genes are present or not, but major effect is just happening because of the small s, this recessive gene present. So this spotting gene S is a major gene controlling the spotting, while modifying genes are minor genes affecting this trait. So this is the major gene which is affecting the color spotting, while modifying the genes are the minor genes affecting this trait. So which are affecting this trait, they are called as minor genes in this case. So that's the difference between your major and minor genes in this case. Then comes the point of inheritance of quantitative characters. So guys, these now we are going to the some more advanced version of your genetics today. So be with me. So we will go through some different terms which were asked in exams most of the times, or in the oral interviews. So like what are concept of polygenes? So color, sex, etc., which shows distinct categories are known as qualitative characters. So they are qualitative characters. So they are usually governed by one or major genes or oligogenes. Uh, so characters like length of ear in corn, yield of grain, yield of milk, stature, etc. They are all come under the less continuous variations, and they have large number of minor genes called multiple genes or polygenes. So characteristic features of quantitative characters is they are continuous variations and marker influenced by the environment on the expression. So we will deal with this continuous variation and how the environment is affecting with the difference part of qualitative and quantitative characteristics in upcoming lecture also in more details. Then comes your transgressive segregation. This is the appearance in the F2 individuals with the higher or lower intensity of characters. Then the parent is called transgressive segregation. So if you have higher or lower intensity of your character than the parent, then that generation that you have produced in the F2 individual, that will be called as transgressive segregation. It is produced when the parents have positive alleles of different genes affecting a quantitative trait, and segregation of these genes produce two extreme homozygotes in F2 which transgress the parental limit for the character. The reappearance of ancestor is called atavism, throwback of reversion. Now comes a term called penetrance, and then we will go for the term polydactyl and then expressivity. So these three terms are quite different from each other, but quite important also. So what is penetrance is? Penetrance is the frequency with which gene produces a phenotypic effect. Just that is the only the frequency with which gene produces a phenotypic effect or visible effect in the individual, which carry it and is known as penetrance. In other words, we can also say that penetrance refers to the proportion of individuals which exhibit phenotypic effect of a specific gene carried by them. So in general, genes express themselves in all individuals in which they are present in the appropriate genotype is known as penetrance. It indicates the number of individuals that give expected phenotype to any degree. Then polydactyl is the extra finger or toe in human. So like Hrithik Roshan, he has he has extra thumb actually, a joint thumb. So that is happening because of the polydactyl gene present. So there is a presence of dominant gene called P. Because of that, you have this kind of expression is happening. So the normal condition is produced by the genotype capital PP, and the genotypic and the capital PP uh, produces polydactyl conditions. So some heterozygous individuals are not polydactyl like capital P small p. Therefore, gene penetrance of so the frequency of gene produces that phenotype is less than 100% is said to be incomplete penetrant. Yeah, so that's how the penetrant terms come here. And a gene through penetrant may be quite variable in its expressions. And the degree of expression produced by penetrant genotype is termed as expressivity. This term is known as expressivity. Uh, so last term that we were discussing expressivity is a degree of phenotypic expression of penetrate gene is called how much of phenotypic expression of your penetrate gene that you're looking for is called expressivity or ability of gene to produce identical phenotypes in all individuals carrying it in the appropriate genotype is known as incomplete expressivity. Many genes have incomplete expressivity while the wild type alleles buffered against such variations. So that's it for this lecture. So now comes the next lecture for you, qualitative. And because we were discussing the last part as qualitative and quantitative characters, so we continue the same lecture with the next part. Then we will take some break afterwards that. So this is the next part of your qualitative and quantitative trait. So what are qualitative genetics? What is qualitative characteristics? What are qualitative traits? So in the case of inheritance of multiple genes, The phenotypic traits of different organisms may be of two kinds. They could be qualitative and quantitative. The qualitative traits are the one which are in the classical Mendelian structures like form, structure, pigment, antigens, and antibodies. These all includes your qualitative. We see the quality of your characteristics. So like they are either horn or hornless, round or wrinkle seed, black or white guinea pigs, or blood group type of mans, AB or AG. So that's how we see the mainly the qualitative traits in that case. So each qualitative trait may be under genetic control of two or many alleles of single gene with little or no environmental modifications to obscure the gene effects. And the organism possessing these qualitative traits have distinct phenotypic classes and are said to have discontinuous variations. These qualitative traits have distinct phenotypic classes. They are separate from each other and they are said to have because of that they are called as discontinuous variations. They have discontinuous variation in the qualitative characteristics. On the other hand, your quantitative traits are the one which are economically quite important, such as height, weight, skin pigmentation, amount of flowers, fruit, seeds, milk, meat produced by plants or animals. So all these includes your quantitative genetics. So these are called part of quantitative traits. So they are also called as metric traits because we are varying in the quantity form. Uh, uh, finding their what we say their trait in the metric form, so that's why we call them metric trait, and they do not show clear cut differences between individuals and form of spectrum phenotypes which blend imperceptibly from one type to another cause continuous variations. So such genes which are non-allelic and have phenotype of single quantitative trait are called polygenes or cumulative genes. So inheritance of polygenes quantitative trait is called quantitative inheritance, multiple factor inheritance or multiple gene inheritance, or we also can say polygenic inheritance. So all these term comes under your quantitative inheritance. And genetic studies of qualitative traits is called as qualitative genetics. Same hold for if somebody is studying about quantitative traits, the study of quantitative traits is called as quantitative genetics. So these are the certain characteristics of quantitative inheritance. That is, quantitative inheritance are following characteristics. The segregation phenomena occurs at an indefinitely large number of gene loci. Um, Substitution of allele occurs in gene locals, then such as allelic substitutions have travel effects. The genes for the multiple traits have different biochemical functions, but the similar phenotypic effects. Therefore, phenotypic effect of gene substitutions are interchangeable. And polygenes have pleiotropic effect, that is, they may modify or suppress more than one phenotypic trait. So that is known as polytropic effect. And single allele may do only these things chemically, but ultimately many affect many characteristics. And environmental conditions have nave considerably the effect of phenotypic expression of polygenes for quantitative traits. So for like height is genetically controlled quantitative trait, but some environmental factors such as soil, fertility, texture, all they might affect the height also. So environment could play a role also in the case of quantitative genetics. So, for example of quantitative inheritance, there is a kernel color in wheat, that is Nielsen and Abel and East. They did their first experiments on the quantitative inheritance by working their individual work on the wheats. They did separately their work, and what they did is they crossed between the red kernel and the white kernel, yeah, of wheat plant, and grain from the F1 was uniformly red, and but of the shade intermediate between the red and white of the red and white of the parental generation. So they saw incomplete dominance, but when the F1 offsprings were crossed again themselves, so F2 generation shows five different phenotype classes. That is in the ratio 1:4:6:4:1. So that was surprising. So in the first case, when they mixed with the red with the white, so they saw a mixture of medium red color. Okay, so that there was incomplete dominance was happening, like in the yesterday we study with the red and white we saw a different color. But then when they did the F2 generation, they saw that there was a dark red, red, medium red, light red, and white. So that was a bell-shaped curve was coming up. So that's happening in the always is happening in the quantitative genetics. So they said that, noting that there's a 1 is to 16 of the F2 was extreme in color, and either of the parental plants, they theorized two pairs of genes controlling the production of red pigment. So they say mainly the two things were playing that role. Two alleles were playing that role, and they were capital A and capital B. And alleles resulting in the lack of pigments. Red was happening because of capital and capital B, and the lack of pigment was happening because of the small and small b. So they saw this result as 1 is to 16 red, 4 is to 16 dark, 6 is to 16 medium, 4 is to 16 light red, and then 1 is to 16 as a white one. So this was the cross of them. So when they have first in the red and white, they made a cross. So they have medium cross, capital S small a, capital B small b, like this one, and then this cross looks like this, right? And then when they made this Punnett scare, they saw that there's a red, totally red, and then there's a middle red, dark red, medium red, and light red also, and then there's a white also, and that was all happening because of the epistatic behavior of these genes. So there's another example in the quantitative genetics, is that another classical example of polygenic inheritance was given by Davenport in Jamaica, who studied about two pairs of genes, that is capital N small a, capital B and small v, causing skin pigmentation between Negro and Caucasian people. So there's a dark people and Caucasian people. So because they were happening because of the capital A and small a, and capital B and small b. So these genes were found to affect the characteristics in the additive fashions. Thus, Negro has four dominant genes, that is capital A, capital B, and white one has four recessive genes, small a and small b. So F1 offspring, when they cross between each of them, uh, mating of capital small a and capital A, these dominant genes and recessive genes, all are capital A as small a, capital B small b, and they have a mulatto skin color, mixture of both white and dark. And these mating of two such mulattoes produces wide variety of skin colors, and they also produces wide variety of skin colors ranging from a dark skin to the white skin, and the results looks like this. You have a negro, then you have a white, you have a mulatto, middle one, you made a cross, and then your phenotypic ratios come out to be with the black negro to be 1, dark to be present 4, intermediate to be present 6, light to be present 4, and white to be present 1. So we also see in this quantitative genetics a bell-shaped curve, and this is the genotypic frequency written here, like this one. So these results are clearly showing that capital A and capital B genes produces about the same amount of darkening of the skin. Therefore, increase or decrease of capital A and capital B genes cause variable phenotypes in the F2 ratio, in the ratio of 1 is to 4 is to 6 is to 4 is to 1, like in this case. Yeah. So this and another example in the quantitative genetics is the skin color we have discussed, which is an example of polygenic inheritance because of two pairs of genes were present, and these two genes were making a polygenic inheritance in that case. So further, inheritance of height in men is a complex phenomena which involves 10 or more pairs of genes in that case. So characteristics of tallness is recessive to shortness. So tallness is dominant, recessive is shortness. Thus, an individual genotype of more dominant genes will have the phenotype of the shortness. So because the quantitative trait is controlled by multiple pair of genes, we have and influenced by the variety of environmental conditions, we have a range of 140 centimeter to 203 centimeter. So in public we have 140 centimeter to 203 centimeter of heights. So this is also somebody made this bell-shaped curve, and you can see the with the quantitative inheritance height in inches. This is the height in inches, and this is the number of males. So you can see that how it is being making a bell-shaped curve in the society also. So that's the beauty of genetics in the quantitative characteristics. Some another example from seashells, you can see it. Then we have discussed an intense amount of your quantitative characteristics, and we have understood many parts of it, how things are working out. Let's continue now with the other part, that is qualitative trait characteristics. So easiest characteristics or traits to deal with those involving discontinuous or qualitative trait differences that are governed by one of your major genes. Examples are starchy versus sugar kernels, or determinate versus intermediate habits of growth in green beans. So these are all examples of this qualitative traits. And qualitative trait is expressed qualitatively, which means the phenotype falls into different categories. These categories do not necessarily have certain order, but they have inheritance of qualitative trait, that is monogenetic, which means the trait is only influenced by single gene, or inherited disease caused by the single mutation are good example of qualitative traits. Another is blood type. So all these are the various example of qualitative traits. Environment in this case in the qualitative characteristics have very little influence on the phenotypic of these characteristics. So environment does not play a big role in these cases. So these are the major differences between the two, qualitative genetics and quantitative genetics. So qualitative genetics is deal with the inheritance of the trait of a kind, that is form, structure, color, where this one is the deals with the inheritance of the trait of degree. How much degree is the trait of your inheritance? It's like height, length, weight, number. This one is discrete phenotypic classes occur which displayed continuous discontinuous variation, but in this case we saw continuous variation. This is governed by two or many alleles of single gene, but in this case it's governed by non-allelic genes or polygenes. Here the phenotypic expression of gene is not influenced by environment, but here it's highly influenced by the environment, your polygenes. And it concerns with the individual matings and their progeny, and it's concerned with the populations of organisms consisting of all possible kinds of mating. In it, analysis is made by counts and ratios. In it, analysis is made by statistical methods. That was the major difference between your qualitative and quantitative traits. So there is one some example is there. So like in qualitative trait, you can choose blood and serum type, the enzyme defects. So mode of inheritance is single Mendelian is happening, and environmental effects is little in that case in the qualitative part. So in the quantitative grade, you can check height, intelligence, milk yield, all matters. So mode of inheritance is complex, it's a polygene, and environmental effects is moderate to great heights. So this is how the graph of this normal and disease looks like to a bar graph. Normal looks like this much, disease looks like this much, but in the case of quantitative you will always see a bar shaped curve like this. And this is a threshold is showing us the third part, moderate to great, like how number of disease are being affected in that case with the frequency. So that was the major differences between qualitative and quantitative traits with some examples shown to you. So I think we are done with the two basics topics. So now we can have it a little break here. Any questions, students? Okay, you can think of your questions. I come at 9 then. So we start with our last lectures of multiple factor hypothesis, where we will study about how that hypothesis came and how it could be useful in our genetics part also. So I see you in 10 minutes then. So, students, I'm back. Um, let's continue the rest of the stuff. How assignments are supposed to be done. I share with you some example. Or so I taught somebody in my last lectures about marker assisted breeding. So they made advantages. What are the different types? And just in one page, she has finished. And took some figures from it. So all the important parts that you think from these sheets that you required, you're allowed to use it to make it happen. This was a good one. So somebody made this one. This was very nice. Like you can see, all concepts, all everything is covered with the history, maintenance, law of inheritance, test process. Everything is covered very nicely. So I can share with one of you students in the WhatsApp group so that uh, you can think that how assignments looks like. Where does it download it now? This one. So I share in the group. I really like this one. So you can see an example how assignment is done. So I will continue with the last topic now. I hope your query is clear, Gauri. So this will not take much of my time, but this was essential part to continue because this is dealing with the qualitative and quantitative traits. So I want to do it in continuity of this lecture because it will be very bad if I don't make this done. So that's why I continue with this. So multiple factors. So it is a quite natural that small differences exist among the individuals of similar genotype due to the effect of environmental on the genotype. On the other hand, there are some heritable differences also exist the continuous variation. Most of the economical traits show continuous variations and they are measurable or quantifiable regions. So quantitative characteristics, as we know, they show continuous variations. They have multiple genes, multiple factors, polymeric genes, and inheritance shows same Mendelian principles. Whereas the qualitative characteristics shows discontinuous and governed by one major or two major of oligogenes. We have discussed an ample amount of about the qualitative and quantitative characteristics in our previous lecture. Multiple factor hypothesis was given by again these people, Nilsson and Ahle. We have discussed with the kernel color with the quantitative characteristics that he showed that the red color kernel wheat is produced by capital R, with the true breeding with the F1 generation producing capital R small r and F2 are giving red and white, indicating a dominance of red over white. However, with the careful examinations, what they saw that F1 red was not intense as of the parents, and F2 has observed two grades of red in that color. We have discussed that already. And some crosses ratio of 15 red is to 1 white ratio was found in F2, indicating that there were two pairs of genes. And finally, he observed different shades of red in F2, showed red shades white as follows. So we have discussed this in previous lectures very nicely, red and white color, red, wheat and white color, and giving this red, dark, medium, light red, white one. So that is your multiple factor hypothesis. So uh, uh, it was concluded two duplicate dominant alleles, R1 and R2 cumulatively decides, and R1 and R2 are completely dominant over white in this case, and their highly intensity of red color depends on the number. So that's a Punnett scale method showing you how it is done. Then there are also genotypic ratios, why they were dark red, medium red, light red were happening. So hence, if two parents differ for the two genes, so aggregations of 1 is to 4 is to 6 is to 4 is to 1, provided both R1 and R2 contribute equally to the color. So if three genes are involved in F2, segregations, if like this case we discuss about two genes, but if it is three genes are present, so then the segregation of your genes would be 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1, like this. For red shades and one for white. So like this. So Nelson LA multiple factor hypothesis states three major terms. That is, for a given quantitative trait, there should be several genes. For the single trait, there should be quantitative trait. There should be several genes which are independent in the segregations, but cumulative effect on phenotype. Dominance is here usually incomplete, and each gene contributes something to the strength of the expression of a character, whereas recessive alleles does not of genes present dominance gene, so they are not doing so. So causes of genetic variations, multiple factor hypothesis. So we are used to think about genetics of discrete plant phenotype, for example tall versus short peas in Mendel experiments. So in this example, variations at a single gene contribution to a major phenotypic differences, and the many traits of interest, that is often the case of large number of genes, hold modest effect collectively contribution to the genetic variation, and this is called as your multiple factor hypothesis. So if it is three was the case, like in this case, three independent factors like capital A, capital B, and capital C, with a small b, small c, small a, small b, small c. So in this case, we will see that ratio that we were discussing previously. 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. So there were two general conclusions that came from this multiple factor hypothesis. That is, first, sexual reproduction can produce huge diversity of the genotypes. For example, two alleles with capital and small a can produce three different genotypes. And 10 diallelic loci can produce up to 310 to 60,000 genotypes. Second, this huge potential diversity of genotypes, apparently new types appearing within the population, may be the result of the rare segregants rather than the new mutations. Then this multiple factor hypothesis examined quantitative variations in large number of plants, and they recorded F1 and F2 generations were recorded, and then there was cell fertilized outbreak of seen was in the F2. So like in this case, we can see there's F1 P1 plant and P2 plant with the different length of your cones, the ear size of your cone. So so there are the numbers of cones here written, 3, 11, 12, 15, 16, 7, 2. And here are the second F1 generation from this comes out to be like this, somewhere in the middle. And further, when they have the F2 generations, so they have all from their like length of 7 centimeter to length of 19 centimeter. So in the middle they came out to be. So here they saw a different characteristics, and that is data from the East in 1911. So here your multiple factor hypothesis majorly is there. Why? Because these three different populations show roughly similar amount of variations, and F2 generation shows considerably more variations, reflecting the diversity of genotypes. So we saw a lot of diversity in this case. So that's what the multiple factor hypothesis states that. Okay. So that's it for today's bachcho. That's So I will share this uh, in the group. I hope your all questions are clear today. Rare segregants are the one that we are that's happening in the environment, quite not so frequent, and they are the one which are happen in quite in rare, that are rare segregants. So students, thank you very much. So that's it for today. Uh, we go slowly and slowly like this, not so much pressure on you, because it's get really complicated with each day. So I want to keep it like this. So I shared everything is shared now in the group, so you can go through it. And yeah, till then, keep smiling, stay safe, enjoy your life, and see you soon tomorrow. Same time, same session, same going on. Yeah. Take care till that time. Good night, everyone. Bye bye.